12:05, so cat.
I think I'm recording.
Yes, record, Lawrence. Let's get it started.
All right, let me just share my screen. Okay, can everyone see that? Just like a little thumbs up. Cool. All right, so obviously, yeah, we're going to be doing your revision lecture today. So we didn't do all of the content that you guys did, but we've kind of tried to accumulate what we do know and made lectures for you guys based on what we were all based on. So you might find that the content might be a little bit jumpy. You will be learning some of the last part of your content first in this, which will hopefully just be a revision for you, but we'll upload the slides for you and you guys can check back in, and we'll do questions at the end. So feel free to just pop it in the chat if you'd like to, and we'll answer it towards the end once we've gotten through all the content. And I think that is it. I'm just going to get started. Okay. So hominid evolution. So obviously humans, we used to be bipedal. So meaning we are bipedal now, meaning we walk on two legs now. But when we were great apes, we were quadrupeds. So we basically used four limbs for most of the time. And the reason that we became bipedal is because basically it was advantageous for us to be able to walk on 2 feet. That was to be able to move greater distances and to hunt. It also meant that our hands were free for tool use, so to use knives, spears, flint and stone, things like that. And it also meant that we had much finer motor control. And that's basically the drive for all evolution. It's the main thing for you to remember is that it comes about because of advantage. If it's not advantageous, it's not going to happen, is it? So then we've obviously got these skeletal structure changes. So our big toe became smaller. And then our pelvis became more bowl-shaped, and that was basically to be able to support our weight a lot better. So you can see in this image here, pretty much, the way that our weight is distributed. So it comes straight down the center, and then it lands in our pelvis, and we need to be able to distribute that weight somehow, given we've got 2 legs. And so the way that we're able to do that is because our pelvis is bowl-shaped, and also because our femur is slanted inwards. So that was another evolutionary advantage that occurred, was that our femur kind of bent in, and that's just to direct the weight more towards the center rather than out. So you can kind of see that just here. The other thing that happened was our foramen magnum, which is basically where our spinal cord joins with our skull. That moved more anteriorly. So our anterior is the front. So it began kind of towards the back here, and it moved more towards the front of our body, towards the front of our head and towards the base of our skull. So you can see that one just there. So it's quite posterior there, quite towards the top of our skull. And it basically just moved more anteriorly and also more towards the base of our skull. And then the other thing that happened was we got less robust arms. I saw that in your notes. I'm not really sure why that is, but I'm sure your lecturers could help you out on that one. Okay, so now I'm just going to launch straight into your lungs and airways, asthma, COVID, and some of the drugs around that. So obviously the purpose of our lungs and our airways is to be able to transfer oxygen from our environment into our body. And we use that for cellular respiration, which we'll go over with you guys later on. And it's also to transfer waste products such as carbon dioxide from the blood to the air. That's more or less driven by a concentration gradient between our blood and the air in our lungs. By getting carbon dioxide out of our blood, we also prevent acidosis. So acidosis occurs when we have too much carbon dioxide in our bloodstream. So it's basically our pH is too low. And when that happens, we begin to exhale more carbon dioxide. So you'll notice that we start to breathe a bit heavier or breathe a bit harder, or more frequently as well. And then you can see in this image here, we begin with our trachea, and it basically comes down to about here, and then it bifurcates into our 2 main bronchi, and then it goes into our secondary bronchi, our tertiary bronchi, our bronchioles, and then our alveoli. We have 1 main bronchus for each lung, and our right main bronchus is much wider. So you can kind of see it in this image here, actually. So our right main bronchus is much wider. It's actually quite a bit shorter. So it bifurcates here, whereas the left bronchi bifurcates here. And then we also can see that it's much more vertical. So rather than being splayed kind of outwards, it's splayed downwards. And one of the main implications of that is that we actually end up with a lot more infection in our right lung because it's a lot easier for bacteria to get in, given that it's more vertical and that it's wider. And then the other note to make is that gas exchange occurs at our alveoli. And I'd also just like to point out that kind of towards the top of our trachea, or towards the top of our airways, we have a lot more cartilage that supports our airways. But as we go down, we begin to have less cartilage and more smooth muscle. So there's a combination of both at any given time. But the further we go down, the more smooth muscle there is, which is really important when we begin to think about asthma, because the smooth muscle is what we can actually control. Okay. So spirometry is basically, it's a way for us to measure lung function. We can test the amount and the speed of air that we're inhaling and exhaling, and it helps us to identify if there's any obstruction in the lung and airways, more or less when it involves respiratory disease, such as asthma or COVID-19. So some important terms, I don't know if you guys need to know this. I noticed that it was briefly in your lecture slides, but I just thought I'd go over it anyway. FEV1, which I noticed was on one of your diagrams, is forced expiratory volume in 1 second. So when we do spirometry, it's an experiment where you breathe normally to set a baseline for a period of time, and then you take a deep breath in, and then you breathe out as hard as you can. And what we can see in this image is that whole process of breathing out as the volume in our lungs gets smaller. So FEV1 is our forced expiratory volume in 1 second, and it's basically how much air can we get out when we breathe out as hard as we can in a period of 1 second. And then FVC is forced vital capacity, and what that is that's the volume of air that we can get out for our whole breath out, our whole exhalation. So whereas FEV1 is that 1 second, FVC is the whole period of time. So I'll show you that on the diagram. FEV1, so we've got our time in seconds here and our volume there. FEV1 is over the duration of 1 second. We begin at 6 litres and we finish at 2 litres. So our FEV1 is 4 litres. And then for our FVC, we begin at 6 litres and we finish all the way over here at 1 litre. So our FVC is 5 litres. And then FEV1 on FVC is just a fraction, just a ratio of those two. I'm not sure how much of that is applicable for you guys, but it's a really good way to measure any kind of respiratory disease. So if they mention lowered FEV1 or lowered FVC, you know that there's going to be some kind of obstruction in the airways. Okay, so one of those respiratory diseases is obviously COVID-19. I'm sure you all know a lot about it. And that's basically, it's a kind of virus with pretty typical genetic features for a virus. It has a small genome and its nucleic acid is RNA. It replicates via asexual reproduction in the host cells, and it has really, really high mutation rates. And again, those mutation rates, obviously they occur because it's advantageous, which is what we were talking about before. You know, Delta, it travels a little faster, and that it's advantageous for the virus to travel faster because it can infect more host cells and survive for longer. It transmits via droplets, which is why we have the necessity for social distancing and we do such intense contact tracing, is because those droplets, which can actually not just occur when we cough or anything or sneeze, it occurs as well when we just breathe out. So that's why we have the importance of masks and social distancing, because those droplets stay in a room for a very long time, up to a few hours even. So that's why there's such intense contact tracing at the moment as a method to try to manage that. It's considered an obstructive disease of the small airways, so more towards our bronchioles and our alveoli, so much further down at the bottom of our airways.
I'm gonna have to pull you up here. It's a restrictive disease, not obstructive. That's the whole workshop. Okay. But it's it is a lot of similarities with obstructive diseases like asthma and another one we learned about called COPD. But it's primarily to do with the alveoli, the exchange surfaces, right? And you're gonna have like buildup of fluid there. It's going to cause all sorts of gas exchange issues, but it's less to do with the conducting airways, like higher up, not the actual gas exchange, but where the air passes in order to get there. Like that's more asthma, less COVID. But anyway, continue.
Okay, there you go. So if you were taking down word for word what that said, change obstructive to restrictive. So it causes bronchoconstriction, which is basically tightening of the airways, and emphysema, which is shortness of breath due to alveoli damage. So that's kind of what Stone was talking about, was there's a real issue with gas exchange because there's damage to the alveoli. And there's also can be issues with damage to lung tissue. It makes it less elastic, which I'm sure you can imagine if you're breathing in and out is a pretty elastic process. So the less elastic it is, the harder it is to breathe. It's involved with a really huge immune and inflammatory response, and it causes a cytokine storm. I'm sure most of you guys know cytokines are really important mediators of an immune response, and we'll come to that a little bit later. But more or less what happens is it just makes it really difficult for oxygen to get in, and it lowers FEV1 and FVC. And an important note to make is that we don't use spirometry in COVID-19 because you have to breathe out really hard. And an issue with a disease that's transmitted by droplets is breathing out really hard. So we don't want to make a dangerous environment by asking people to do this. And I thought I'd quickly just outline this asexual reproduction here. I'm not sure if you guys have to go over it, but basically what happens is that the virus binds to our ACE2 receptors, which is an endogenous receptor, and it's endocytosed, and the viral capsid reveals the viral RNA, and it uses the host machinery to replicate, produce its own proteins, goes through this whole process, and basically it's just released so that it can infect more cells. And that's how it replicates within our own system.
Can I quickly jump in here, Kat, again?
Mm-hmm.
Go back one slide, yeah. Just really quickly, you guys, I'm not sure if you had this or not, but when we did this, there was a big focus on the inflammatory host response versus the actual virus causing damage. So there's two mechanisms in which your body can be damaged. In the first phase, it's the kind of viral spread. We kind of target that with antiviral drugs. But in the later phase of disease, in like the severe, ICU hospital cases, a lot of the time it's to do with an overreaction of inflammatory responses. And so yeah, you get an excessive cytokine storm, you get excessive immune response attacking yourself. And that's the latter, late stage part of the disease. And in that case, we treat it using more anti-inflammatory rather than antiviral drugs. But anyway, that's
I'll get to as well. Okay, so asthma is our other respiratory disease that we spoke about. And it's a disease that's primarily one of chronic inflammation. And so you guys discussed that there are two types of asthma. There's allergic asthma and there's non-allergic asthma. Allergic asthma tends to be more early onset, and you actually tend to see it in a more male population. And then non-allergic asthma tends to be more late onset. So it causes inflammation, obstruction, so bronchoconstriction, and hyperresponsiveness. And yeah, it leads to obstruction, reduced airflow and difficulty breathing, and lowered FEV1, which is, like we spoke about before, a difficulty in breathing out. So you can really see here like that really intense inflammatory response. And you can see how it would restrict our ability to breathe. And then also here's just a mucus plug, which has occurred because of the inflammatory and immune response. So our allergic asthma pathogenesis. So in our airways, we're constantly being exposed to antigens, some of which are pathogens, some of which are allergens. Our allergens are presented by antigen-presenting cells, so APCs, to naive T cells. We then respond to the antigen by differentiating into T helper cells and releasing IL-4 and IL-3, which are some of those cytokines we spoke about before, which then stimulate B lymphocytes. These B lymphocytes then produce IgE, which is a type of antibody, which is primarily involved in allergy, and they bind to and prime mast cells on the Fc epsilon receptor. Okay, so on next exposure, once these mast cells are primed, they degranulate and they release immune mediators such as histamines, which cause vascular leakage, edema, which is like flooding of the alveoli. Again, that's what Stone spoke about before. Bronchospasm and airway obstruction. And it also recruits, I'm not 100% sure how to say this, eosinophils, which are prime mediators of inflammation. And then you've got eosinophilic and non-eosinophilic asthma. And you can see some of the criteria for that over here. I'm not going to go through that. All right, so I just wanted to go through some of the immune cells that I mentioned there. So mast cells release granules containing histamines and some other active agents as well. And we also spoke about briefly some antibodies. Now, antibodies come in a few different isotypes. So you have IgD, IgA, IgM, IgG, and IgE. And IgE is the main one we spoke about. You have a heavy chain, which you can see in green here, and a light chain. And you've got disulfide bridges holding it all together. If you want to see a little bit more information about that, it's kind of in the diagram there, but basically there's the FC region here determines the isotype. So whether it's going to be IgE or IgM or whatever. So mast cells and antibodies, they're both obviously useful. They're there for a reason. We do use them in our normal immune responses, but they can become overactivated. And that's when they really become an issue. That's when we see a lot of disease. And then, yeah, allergies are becoming more and more prevalent due to increased hygiene. So at a young age, we're being exposed to a lot less dirt and allergens and things like that. Pathogens, sorry. And at the young age, when our immune system tends to be kind of learning and adapting. And damage to the epithelial barrier leads to an increased exposure to pathogens. And I just wanted to outline here the difference between asthma and COVID-19. So asthma kind of occurs at this level here. So like our bronchioles and then some of our larger bronchi. And then COVID-19 is much more at our exchange surface. So the much lower end of our bronchioles and our alveoli. Okay. Before I move on, so I was just going to quickly outline the difference between agonists and antagonists. So this is, I pulled this from your lectures mostly, but agonists stimulate the receptor protein function. So when you release an agonist into the system, if it binds to the receptor, it's going to do what that receptor is supposed to do. It's going to encourage the receptor to carry out its response, whereas antagonists will prevent any receptor function. So when you have an agonist on its own, you'll have a full response, which is like a bit of a dodgy way to say it because you can have agonists that are a little bit less potent, that aren't quite as strong, but basically you'll get a full response. When you have an agonist and an antagonist, they'll compete for the same receptor site. And if it's a competitive antagonist, you'll be able to actually overcome that by putting more of the agonist into the system, but you need a lot more concentration, so the drug becomes a lot less potent. And then with an antagonist, you'll see no response at all. And so this is just a quick outline of one of the, I think this is the main agonist that you spoke about, is adrenaline as an endogenous agonist for beta-2 adrenoceptor. So beta-2 adrenoceptors are located primarily in our smooth muscle, and where you guys are concerned, we're speaking about the bronchi and the airways. And so like I spoke about before, there's quite a bit of smooth muscle in the airways. And one of the main things that the beta-2 adrenoceptor does is that when it's agonized, so when we're putting something like adrenaline into the system, it relaxes the airways, so it widens them, which would be advantageous because when we're considering our sympathetic system, it's stress, so sympathetic and stress are kind of, you can intertwine them. And if you're stressed, if you're trying to breathe harder, work faster, you want to be able to get more oxygen into your blood, basically. So that's why we want to widen our airways. So when we're considering drugs to treat asthma, some of our first options are beta-2 adrenoceptor agonists, which cause bronchodilation, which is what I was talking about, is that relaxation of the smooth muscle. Now, these can be long-acting, they can be short-acting, but they should be inhaled to avoid any adverse effects, because like I discussed, beta-2 adrenoceptors aren't just in the airways, they're actually in a few other places. So if we were to ingest them into our whole system, it would have a lot of adverse systemic effects that we don't want. So an example of these is salbutamol, terbutaline. I'm not going to go through them all, actually. I don't want to embarrass myself. And then we have the glucocorticoids, which as a quick note can actually also be used in the treatment of COVID-19, more towards the end of the disease, though, like Stone said, not at the beginning. We don't want to administer it first. At the start, we want to administer an antiviral drug instead. But once it gets a bit worse, we can use dexamethasone. So yeah, they're used to reduce the inflammatory response. And the reason that we don't use it in COVID-19 at the start is, and we tend to just use it for more severe cases as well, is because it acts on cytokines. So actually, I'll ask you guys, you can put it in the chat if you'd like to. If we're using it to try to reduce a cytokine storm when it's causing more damage than good, why might we just want to use it towards the end and not at the start? And you don't have to say anything, but please do. Don't leave me hanging. Okay. Yeah, so basically it's to give our body a chance to fight the virus, and it causes a little bit of harm. So at the beginning, with any kind of infection, we want to actually give our immune system a chance to do what it's supposed to do. It's usually pretty good at its job. So we just kind of want to give it a chance to work its magic, I guess. We might administer it towards the end because that's when we have too many cytokines, too much immune response, and that's causing that excess inflammation that really makes the disease quite brutal and damaging. So yeah, in COVID-19, we used dexamethasone, and in asthma, we tend to use these other ones, and they just reduce the inflammatory response. And I just want to note as well that in asthma, we actually use beta-2 adrenoceptor agonists and glucocorticoids at the same time because we want to combat both kind of symptoms of the disease, which is that inflammatory response, but also the bronchoconstriction. And they should always be taken at the same time. And they're generally inhaled too.
Sorry, Catherine, I have a really quick question. Would the glucocorticoids, oh, sorry, I'm sorry if I mispronounced that. Is it an immunosuppressor?
Yeah, pretty much.
Okay, cool.
Yeah. Okay. And so then this is more what you guys learned about. You learned about using monoclonal antibodies to treat asthma. We tend to only use monoclonal antibodies to treat really advanced asthma, really when nothing else has worked, basically. So we have monoclonal antibodies. They have this suffix of UMAB. So if you see UMAB, you know that it's an antibody. And they can restrict all kind of different parts of asthma. So I spoke about the process kind of briefly before. I think I put this diagram up. I hope I did. And we have all these different mediators. So we have these cytokines, we have the IgE, and these different monoclonal antibodies target different parts of that immune response to try to dampen the response, basically, and just lessen its intensity. But we tend to not want to use these because, like we kind of spoke about with glucocorticoids, anytime that you use something that targets a mediator really broadly can have some adverse effects that we tend to want to avoid unless it's a really severe case. And then the last thing I was going to go over quickly was just this idea of good medicine. So we have these that mediate it for us. In the USA, it's the FDA, which I'm sure you guys have heard of. And in Australia, it's the TGA. And basically, these are introduced to try to regulate manufacturing, sale, and advertising of medicine, make sure that it's safe, making sure that it has a high efficacy. So efficacy being that it actually targets what we say it's going to target, and that it's got really good manufacturing. And the reason that we want it to be manufactured properly is because it could have infectious material on it. So for example, in like 1902, about 19 children died of diphtheria because they got an immunization, some children, died after they got the diphtheria immunization because it was infected with tetanus. So that's the kind of thing that you want to be able to avoid. And then the other thing is, I've got another example here of when you want to test properly. In Kansas in like 1937, there was a chemist who tried to alter a drug, basically to turn it into a liquid. And to do that, he used this substance that actually consequently caused about 100 people to die. And the reason that he was able to do that was because of the lack of restrictions around it, the lack of testing. There was no clinical trials or anything like that. So a lot of people died because they didn't understand the drug interactions that it would cause or the way that it would interact in the body. They hadn't put any of this on any kind of model before letting humans have it. And then the discovery of drugs, it can happen two ways. You can kind of do it based on, okay, well, I know that this disease begins because of this target here. So maybe we were looking at beta-2 adrenoceptors, for example, and then we tried to think about ways that we could agonize those receptors. The other thing that you might do is you might go, okay, well, I know that this drug helps the disease, but I don't know why. So I'm going to try to figure out what the target is and develop a drug to target it a bit better. So there are kind of 5 phases of development that you can read there. I'm not going to go through. And then I just had a last slide for like tips for your exams, but I feel like we might do that more towards the end. So I'll just leave that for now. And I will let someone else do theirs now.
Thank you very much, Kat. Again, sorry for interrupting you at the start, but also just want to say again, thank you for doing that, because like we've finished our exams, and Catherine here has taken her time out of her day to revise some of that for you guys. I hope you find it useful. Lawrence, because like you have to go as well from yours.
Yeah.
Right? So do you want to go ahead?
You want me to go next?
Yeah.
Okay, let me. Give me a second. What do I even press? Okay. Can everyone see that? Can you guys see that? Okay. All right. So I'm Lawrence, and these are the kind of topics that we're going to go through today. So the first one is mainly about like bacteria and mitochondria. The second one is the endosymbiotic theory, which kind of is related to bacteria and mitochondria. And then we're going through cellular respiration, which, yeah, everyone hates. We legit have to learn it every year. And then we're going through Heine-Weinberg equilibrium. That's not too bad. We're going to go through like an example of that. And then the last 2 topics are mostly about like immunity. And it's somewhat related, kind of related to what Kat did with asthma and COVID-19. Okay, so to start off, here's kind of like a basic summary of the differences and the similarities between mitochondria and bacteria. So bacteria is a eukaryote, and mitochondria is a organelle that's inside of a eukaryote. So both of them have ribosomes, and mitochondria have ribosomes that resemble much more like bacteria than our own host cells. And so there's actually like some kind of implications when we use antibiotics. Some of the antibiotics that we use actually would influence on, to have an influence on mitochondria. So that's kind of like your side. So that's where your antibiotic side effects kind of come from. So they both have a circular genome. I'm sure you've heard about mitochondrial DNA. It's a bit smaller than bacteria, and yeah, they're both circular. Bacteria also have these little extra chromosomal bits of DNA, which are known as plasmids. Not all bacteria will have them. So yeah, they both have protein transport systems, and that's because they've got membranes to pass through. So we'll go through like these ones. So Sam, Oxa, Tim, and Tom. And then they both do binary fission. They both have membrane folds. So the membrane folds that we see in mitochondria are to do with the inner membrane. So the outer membrane is like this like normal circular capsule shape, whereas the inner membrane is more folded and more like convoluted, really. And this is important because when we get to cellular respiration, it's important because it increases the surface area for us to do for the mitochondria to do that aerobic respiration. Double membrane. So yeah, like mitochondria has an outer one and an inner one, whereas the bacteria, only gram negatives. I'm not sure if you've learned about gram negatives before, but gram negatives is it's a type of, it's like a category of bacteria. And more specifically, they have 2 membranes, and they also have like a little thin cell wall in between it. Whereas on the other side is gram positives, and those only have a single membrane, but they have a really thick cell wall layer. Right. So endosymbiotic theory. Pretty much, like if I wanted to summarize this, it's pretty much just Pac-Man, bro. Like it's just So we originally had an ancestral eukaryote, and we had a aerobic bacteria, and literally the ancestral prokaryote ate the bacteria that could do aerobic respiration. And then somehow they didn't kill each other, and then they developed what we call a symbiotic relationship. So usually these ancestral prokaryotes or eukaryotes would hunt other bacteria down to get nutrients and literally like just digest the bacteria for growth and nutrients. So they're nucleated and they can only do glycolysis only before. So there was no electron transport chain or anything like that involved. These ancestral species were most likely facultative anaerobic. So they basically became facultative anaerobic once they gained the bacteria, the mitochondria, or when they ate the bacteria. And and so basically facultative anaerobic means that they can do both aerobic or anaerobic. But they prefer to do aerobic respiration if there's oxygen. So it's like preferentially aerobic, but they can do anaerobic as well, like us. So when the ancestral cell species ate the aerobic bacteria, it kind of used it as like an energy farm where the ancestral cell would supply glucose to this bacteria, and the bacteria was able to kind of produce energy out of it and supply it to the cell. And so in terms of the symbiotic relationships, in terms of both kind of parties were able to have an advantage, really. So for the bacteria, the one that got engulfed, like eaten up, it was able to survive. It was kept alive, and it was well protected around its surroundings. And it was also in like this new environment inside of this ancestral kind of prokaryote or eukaryote. And so there was no competition, and it could still replicate freely, and it could still pass on its genes and evolve, basically. And in terms of the eukaryote or the ancestral cell, it was benefiting from the energy, the efficient use of glucose to make energy. And this is probably one of the big reasons why we've got multicellular life and complexity, because of this efficient generation of ATP. There are these other theories here, which I'm probably not going to go over, but yeah. Okay, so mitochondrial transport proteins. So basically we use these are kind of like protein carriers that are they're embedded in the membranes. And so these are just some of the examples here. I've also on the left-hand side, I've basically listed the mitochondrial, the types that are on different membranes and the most likely direction they transport stuff in. So if you had a protein here, you'd have to go through two membranes. So you technically would have to go through the first one and then the second one. And if you're on the inside of the mitochondria, so the matrix, you would have to go also through two membranes. Usually the proteins that either stem from the mitochondria. So the mitochondria can replicate by itself, and it also has ribosomes, like we said before. So it's able to produce those polypeptide chains or proteins. And sometimes those mitochondrial proteins, they might have roles that are outside of the mitochondria, and so they need to be able to be pumped out into the host space or the cytosol. And it's also the same for the host proteins. Some of the host proteins might also need to go into the matrix. For example, not utilizing these, there are also these transport proteins, but for example, as an example, we need to have pyruvate that's produced at the end of glycolysis from the cytosol to enter the mitochondria. And those use a different, they're not these proteins, but they use a different sort of transport proteins to get into the matrix, where they're able to do OXPHOS, or like aerobic oxidative phosphorylation. And obviously, if you have any like defects or mutations, that's when we get certain mitochondrial diseases. And these transport proteins are basically produced by the mitochondria themselves. So these are derived from the mitochondria, not from the host. All right, so we're going to go through cellular respiration. Yeah, literally every single year I have to relearn this, but pretty much just the basics. We just need to know the basics of aerobic respiration as well as fermentation. And I kind of want you guys to know the inputs and outputs of each kind of like big step. So we've got glycolysis, we've got anaerobic fermentation, we've got pyruvate oxidation, citric acid cycle. So these two are often the pyruvate oxidation and the citric acid cycle, or you might hear it as the Krebs cycle or the TCA cycle. These two are usually like linked together, or they, some lectures might combine it into one. But I've split it up here. And then you've got the electron transport chain, which, makes all the energy. Here's just the top bit is just a really basic overall equation for cellular respiration. It's literally just what's happening from glycolysis to the electron transport chain. And over here, these are just the three main ways that that organisms produce energy. So aerobic cellular respiration basically involves oxygen as the final electron acceptor or the carrier. Anaerobic cellular respiration also goes to the Krebs cycle, but instead of oxygen, they use, those bacteria use there are some bacteria that use other different kinds of chemicals. So things like sulfur and other weird chemicals, they're pretty whack. And we've got anaerobic fermentation, and that's when organisms do not have the ability to undergo the electron transport chain. And so they have to go through fermentation, and there is a reason for that. So principally what all of these stages are trying to do is we start off with glucose, and we want to break the energy down that's stored within this glucose molecule and kind of extract it for the cells to use. And what we do is there are chemical reactions that occur through every single step. And the basis of each chemical reaction is to try and extract energy from the glucose, and we want to store it in the form of redox energy, or just potential energy, basically, on these electron carriers that you might know as NADH, and I think there's FADH over here, FADH2. So we pretty much we have NAD plus, and we load it up to become NADH. And that's what we call a reduction pathway, or like a reduction reaction. And as you know from chemistry, when you have a reduction reaction, you also have to couple it or combine it with an oxidation reaction. And so that's what we're doing to glucose, essentially. We're trying to oxidize it, which is why oxygen's down here as the final electron carrier. And so eventually we load up all of the energy into these electron carriers, and they get shuttled towards the electron transport chain inside of the mitochondria. And what's the purpose of that is to basically unload the redox energy that's stored from these electron carriers and convert it into something that's useful for the cell to generate ATP. And that, what it does is, as it unloads its redox energy through the electron transport chain, at the same time, it's utilizing that energy to pump hydrogen, or H+ hydrogen ions, or what we call protons, from the inside of the mitochondria towards like that intermembrane space that I'll show you later. And basically establishes like a concentration gradient. And we use that proton concentration gradient to drive the production of ATP. The yields might vary depending on textbooks. Some say 32 to 34 or 34 to 38. And that's mostly to do with the fact that different mitochondria will have different transport proteins. And because we need to shuttle that pyruvate into the mitochondria for all of this oxidative phosphorylation to occur, the pushing in of the entry or the import of pyruvate requires energy, basically. It's like it's a mode of active transport, essentially. Okay, glycolysis. So pretty much what's happening is there's 10 steps of chemical reactions, and we're pretty much going from glucose, which is a 6-carbon molecule, into 2 pyruvate molecules, which is 3 carbons. And yeah, just a series of 10 steps. Glycolysis is fundamental for all organisms. So every organism will have some glycolytic ability. And it's kind of counterintuitive because you have to like put in 2 ATP at the start in order to yield or in order to whip out 4 ATP. So we get a net yield of 2. It's kind of like how you have to invest some money into yourself before you can start making money. It's literally like that. And the pretty much the functions of the ATP is to destabilize the glucose so that more energy can be extracted when it becomes pyruvate over here. And when it goes from glucose to pyruvate, it's pretty much like a partial oxidation. So because you've got 2 NADHs being reduced, you've got 2 NAD plus being reduced into 2 NADHs. So it's like partial oxidations. I'm not going to go through the locations and the inputs and outputs. You can go through this in your spare time. Okay, so fermentation. So fermentation is like not the oxidative phosphorylation bit. It's for the anaerobic boys. So basically, we've got pyruvate here, right? And glycolysis. Okay, so so in this step, we're actually utilizing NAD plus, and we're reducing it to NADH. And eventually, if we keep on using glycolysis to generate, our 2 ATP that we get at the net 2 ATP yield that we get at the end, eventually we're actually going to run out of NAD plus. And they're actually more expensive to make than to produce 2 ATPs. So in many of these anaerobic capable organisms, they would undergo something called fermentation, in which they actually reduce pyruvate down to like some kind of other byproducts, whether that might be lactic acid in humans, or in some yeasts, I know they do ethanol, and like carbon dioxide, which is like how your bread works, like baking works. And essentially, the reason they do this reduction is to regenerate the NAD plus. And this is very important because if we don't regenerate this, then we cannot continue glycolysis forever. And if the cells cannot make energy via glycolysis, they're pretty much dead. This is happening inside the cytosol as well. And so for humans, we can do anaerobic respiration, right? But we can't sustain it for a long time. And the reason why is because this step of fermentation and glycolysis itself it produces acid, so H+. And so our muscles will just literally give in, and that's when we have fatigue because of this acid buildup, as well as the lactic acid being produced. Okay, so the next step we're up to is pyruvate oxidation. So that's so this is the start of the oxidative phosphorylation kind of steps. So before we can enter the Krebs cycle or the citric acid cycle, if there's oxygen, the mitochondria will shuttle the pyruvate into the inside, which is the matrix, so the liquid part inside. And basically what it has to do, it has to undergo one step, and to convert it into this molecule called acetyl-CoA, which is it's still 3 carbons. And basically it has to oxidize the pyruvate into acetyl-CoA. And there is, the enzyme is called PDH, which is called pyruvate dehydrogenase complex. And when it does that, it releases some of that energy that's being captured by the NAD+. And this is a very important molecule because it's like a crossroad molecule. It's an intermediate molecule that is produced by not only glucose, but also other metabolic pathways. For example, stuff like turning fatty acids into energy, or proteins into energy. They all all get towards this point, this common step here, acetyl-CoA. And then that's when they enter the Krebs cycle or the TCA cycle. Okay. So this is the Krebs cycle. You obviously don't need to know every single thing here, no enzymes or whatnot. Just know there's 8 steps. And basically we've got the pyruvate or the acetyl-CoA now, and it basically goes around this cycle. And the purpose of this cycle is to kind of release all of the carbons that are left from acetyl-CoA and extract all the remaining energy. So this is, at this step, this is where glucose becomes fully oxidized, and we've got, with all of the energy being loaded up into those electron carriers, which are NAD+, and we've also got some FAD. I just want to note that because, remember, at glycolysis, we've got that glucose molecule that splits into the 2 pyruvate molecules. So because for every 1 glucose, we get 2 pyruvates, it has to go around, 1 glucose molecule has to go around this kind of twice. So we go around this cycle twice because we've got 2 pyruvate molecules or 2 acetyl-CoAs. And so, yeah, and this is pretty much just the inputs and outputs. Maybe I guess you should know the first critical step, which is pretty much, you've got the acetyl-CoA, which is 2 carbons, and we combine it with, with a molecule called oxaloacetate, and that's being regenerated. That will be like kind of regenerated over time through the cycle. And pretty much it's kind of counterintuitive at the start as well. Just like glycolysis, how we invest in 2 ATP at the start, we actually combine the oxaloacetate with acetyl-CoA. So this is a 4 carbon molecule, and we combine it with a 2 carbon molecule, and we form citrate, which is a 6 carbon molecule. So it's like, we kind of like make it bigger and then we kind of reduce it. Okay. And then this is kind of the last step, which is the electron transport chain. Just note that this is occurring in the cristae, which is the inner membrane that's really folded. So this is where the surface area comes to play, is so that we can fit all of these different protein complexes into that inner membrane. And all of these, and these are numbered from 1 to 5. 1 to 4 are used to pump to create that concentration gradient, like I said before, to pump the H plus from the inside liquidy space of the mitochondria to that intermediate kind of gap between the 2 membranes, and to make it really acidic, or because it's full of H plus or low pH. And so what we do is we unload the electron carriers that are in the matrix, and basically they pass through a series of these complexes, and as they do that, they get oxidized, or they get unloaded basically. And using this energy, these proteins are able to drive H plus movement into the intermembrane space. So this is called the intermembrane space. And so by doing so, it creates a chemical gradient, and it's really important. This is kind of like an important concept, and it's all based around the fact that H plus is an ion, and it cannot go across the membrane by itself. It needs some kind of protein carrier or whatnot. So by utilizing that, the fact that H plus is impermeable to the membrane, it can utilize this kind of like, it's what we call potential. It's like potential energy. It's like you having a ball and you're driving it, you're pushing the ball up the hill, and you're just letting it stay there. And so when you let go of the ball, it's going to roll down the hill because of gravity. So it's kind of like this potential energy kind of deal, where we've got this enzyme called ATP synthase, and it's able to make ATP by allowing the hydrogen plus ions that were generated, the concentration gradient. It can use the concentration gradient, basically exploit it, and use that potential energy. So move the H plus ions back down the concentration gradient, back inside. And as it's doing that, it's producing ATP. So and this kind of process is called oxidative phosphorylation. So it's basically using oxygen and these electron carriers, and as well as ATP synthase, to produce lots and lots of ATP. So this is what we call the electron transport chain process. And this ATP process is called chemiosmosis. So that's what we call that process of ATP production, by utilizing chemicals to go down the concentration gradient. So these complexes are named 1 to 5. Just want to highlight here that there's 2 different electron carriers. So we've got NADH and FADH2. Just know that these, they enter at different steps of the cycle. So for example, NADH enters at complex 1, whereas FADH2 enters at complex 2. So they enter at different points, but nevertheless, they still generate the pumping of hydrogen into that intermembrane space. Okay, so chemiosmosis, the last step, with the ATP synthase, basically, using ATP, we're making ATP by driving, by pushing the protons back down the chemical gradient. And how it does is, it's kind of weird. So this protein, this kind of protein complex, it's a transporter of hydrogen. So it allows hydrogen plus H plus ions to diffuse from that really acidic intermembrane space back down into the matrix. And in doing so, it actually rotates. It's like a rotating thing. So it like fills, there's like active sites here for H plus. And as they come down, it does spinning. It basically spins. And this spinning action, this rotational energy, kind of powers the generation of ATP, which is basically getting ADP and then chucking in inorganic phosphate onto it to become ATP. Right. So now we're going to go through Hardy-Weinberg equilibrium. So what do we use this for? Well, basically, if we have the genotype frequencies, or basically the genotypes, if we have a population, just like random people or random organisms that are the same species, and if we count all of their genotypes and traits, we can actually predict what would happen to the genotype frequencies or the allele frequencies. So it's equilibrium. So it's got the word equilibrium in it. So what that means is that the allele frequencies, which is P, and the genotype frequencies, so P squared, 2PQ, and Q squared, those stay constant and they don't really change across the generations. So across each generation of your population. But just remember that just because the allele frequencies and the genotype frequencies don't seem to change across the different generations. So what I mean by the generations is, like, first of all, you have the original population, you count, like, you do your calculations and you count your genotype and allele frequencies, and then they reproduce and then you recount them. If they don't change, that doesn't necessarily mean that it's in Hardy-Weinberg equilibrium. So yeah, just keep in mind of that. So and it's also kind of the reason why we have assumptions or restrictions in place, because in reality, we do, the frequencies do change and everyone is so diverse. In terms of Hardy-Weinberg equilibrium, it's normally used for, basically the model is we've got 2 alleles and 3 phenotypes, and one of the alleles is completely dominant or just basically takes over the expression of the other allele. So that's what we kind of use most of the time for Hardy-Weinberg. Okay, so these are the quick assumptions. So 5 assumptions, pretty much random mating, so that everyone in the population is equally likely to mate with each other, so no one has preferences. No natural selection, so that what that means is everyone is equal in survival fitness, so the relative fitness of everyone is equal. Everyone is equally likely to produce offspring. No mutations. Another caveat to this is that you can have mutations, but only if they're reversible. And if the forward, so like, let's say you've got a mutation A to B and they're reversible, so you can go backwards and forwards. This only works if the rate of the forward mutation is equal to the rate of the backwards mutation. So it's kind of like a chemical reaction, but whatever, I don't think you need to know that too much. No migration is pretty much gene flow. So we can't get new people coming in or old people going out. And we also need a large population. It can be infinite. The best would be infinite, but it has to be large enough so that the allele frequencies aren't affected by just random, by chance. There's going to be variations by chance because evolution is random. So these little slight errors, these random errors, we want to kind of negate that by having a really large population size. So our allele frequencies don't like deviate too much. So this is pretty much, I'm just going to go through a quick example with Hardy-Weinberg equilibrium. So we've got 100 people, and so we've got 40 members that are homozygous dominant, and this would be their genotype B1B1. And we've got 10 members that are heterozygous B1B2, 50 members that are homozygous recessive B2B2. And how do we calculate the allele frequencies for both B1 and B2? So this is pretty much what you would kind of do. So we've got the population numbers here, and this is the total number of animals or people or organisms. So we've got 100 people, 100 things. And so this is the formula to calculate the allele frequencies for B1 and B2. So we basically doubled So whatever number we have here, so the number of homozygous dominant, we double that, and we also add in 10, or the people from the heterozygous, because we double this because they've got 2 alleles, B1, B1, whereas in the heterozygous, we've only got 1 allele, B1 only. And we divide that by, we double this number. So the number of people, we double it by 2. And the reason why we double it by 2 is because every individual, every member will have 2 alleles like that. So that's why it's 200. And then, for the second allele, once you know the first allele, you can easily find the second allele by just doing 1 minus 0, the other allele frequency, or you can do the same calculation. That's because these two must add up to 1. The allele frequencies of both of them must add up to 1 because we don't have any other alleles that can contribute in the population. Okay. And then, so we're going to be testing whether that population with these numbers are in Hardy-Weinberg equilibrium. So the next thing you want to do is, so once you've figured out your allele frequencies, we want to use Hardy-Weinberg equilibrium to predict what do we expect the population numbers to be if it's actually in the equilibrium. So basically, I'm not sure if you can see here, but these are your allele frequencies that we just calculated. And now what we want to do is we want to calculate the expected numbers of each of these kind of categories or phenotypes. And so based off of Hardy-Weinberg equilibrium, we know that P squared equals to P squared plus 2PQ plus Q squared equals to 1. And so we kind of use these formulas here to generate the expected genotype frequencies, okay? And then to get the expected numbers, we basically multiply by the population number. So we've got 100 people, so we multiply each of these expected genotype frequencies by 100, and this is what we expect, and this is what we observe. And now, how do we test this? How do we verify this? How do we statistically show that it is or is not in Hardy-Weinberg equilibrium? We use something called the chi-squared test. It's like a stats thing, but pretty much this is the formula, and we utilize a table like this to observe whether the population is in Hardy-Weinberg equilibrium, and the variations that we see are due to chance, or whether it's 100% not in, it's totally not in Hardy-Weinberg equilibrium. And yeah, so with this chi-squared test, I'm not sure if you've covered this, but we would always have something called a null hypothesis. So we state like an assumption, and we first assume that the population is in Hardy-Weinberg equilibrium. So what that means is we assume that the observed numbers fit in with the expected numbers, so that the differences like 40 and 20, 10 and 50, 50 and 30, the differences that we see here are due to chance, and it's not, and we're in Hardy-Weinberg equilibrium. So that's what we would say for the null hypothesis. And then we want to basically statistically verify whether this is true or false. And we basically use this table. Degrees of freedom, basically, it's always going to be 1 for Hardy. It's basically the number of alleles that we have, which is 2 minus 1. And it's basically it's basically to do with the fact that we have to estimate one variable. We don't have to estimate both of these, because if we know one of them, we can get the other one by doing 1 minus P to get Q, or 1 minus Q to get P. So it's basically number of alleles minus 1. It's basically how many unknown variables did you use or did you estimate? And so basically this is just the maths here. We've got observed minus expected. We square them all, and then we divide by the expected. And then, so this is what we call the chi-squared, and then we add up all of the chi-squareds, and we get 65.3. And then we basically look on this table. As you can see, 65.3 is a bit far. So basically it is significant. So what does that mean? That pretty much means that because it's further, the number's going to be for 65.3, it's going to be smaller than 0.001, which means that our p-value or our probability thing is significant, so that we reject the null hypothesis. And basically what that means is these numbers that we see are not in Hardy-Weinberg equilibrium. Right. So now I just want to go ahead and go through mucosal immunity. to do with the GI tract. So just a brief introduction to the immune system and the GI tract and like microbes in the GI tract. Pretty much our guts are lined by epithelial cells, which are highly specialized because, you've got the villi structure and you've got the microvilli on top of that. And the sole purpose of that is to create a large enough surface area for absorption of nutrients. And we've also got a lot of white blood cells or leukocytes within around these areas, as you can see with these blue dots and this big blue blob here. So, in terms of the immune system, the structures, the tissue structure in the small intestines are basically structured so that they're specialized. So we've got kind of a structure over here, an immune structure called the Peyer's patch. And so this is where you find a high concentration of those immune cells that are involved in T cell and B cells. And you've got other regions of the small intestines that it's what we call the lamina propria, or you've got scattered basically immune cells. And the lamina propria, this word basically refers to anything that's underneath the epithelial cell, the epithelial layer, basically. So you might ask, why is there so many immune cells living around this area? Well, it's because we've got such a large surface area for the absorption of nutrients and food, your body kind of shoots an arrow on itself, where now we've also got a large surface area that's vulnerable to invasion by microbes. And so we need to have lots of immune cells nearby in order to kind of protect these areas. And we've also got, the normal gut microbiota that you might see when you drink your coat or whatever. So those bacteria might also have a chance to invade and infect you as well. So not only do we have microbes, we also have our own healthy gut microbiota. Okay. Branches of the immune system. So this is kind of branching or kind of like tacking onto Kat's presentation. So this is a brief summary of the immune system. It's pretty complex. So we've got the innate immunity side and you've got the adaptive immunity side. So this table is a pretty good summary of the differences and between innate and adaptive. But what I really want you guys to get is that innate is rapid, it's immediate, and it's non-specific, and it doesn't have memory. Whereas adaptive is delayed, it's really strong, it just defends everything, and it can remember everything that it's encountered. And there's a crucial link between these two, and the crucial link are your macrophages and your dendritic cells, because those are what we call antigen presenting cells, and they basically allow communication from the innate to the adaptive. This is pretty much a general process of the immune system, a basic summary of how it works and how the innate immune system links in with the adaptive immune system. So this would be like, for example, your intestines. Let's say you've got the villi and the epithelial cells and the lamina propria area kind of here. And so basically you've got pathogens from the gut, so that you've kind of eaten like contaminated food, and they somehow just infect you, or they just bind to the epithelium, that outer layer, that absorption layer, and they, somehow have a chance to invade you. And so we've got our innate immune cells that are within that lamina propria space that are like, they literally just live there and they're just waiting for people to invade them. And once they sense that they've been invaded, these macrophages and dendritic cells will actually go into the blood vessels or the lymph nodes, and to basically find the T cell or the B cell or those adaptive cells, they have to find the correct one in order to respond appropriately to that pathogen. So the adaptive immunity is specific to the pathogen, whereas the innate immunity is just whatever, it just defends against everything. Okay, so this is kind of just so basically what this slide is showing you is that the GI tract, the immune cells in the GI tract, they need to be in some kind of an equilibrium or a balance between overactivation, so kind of much to do with, you know, how COVID causes that cytokine storm and it causes that over activated inflammation that we see in those severe cases of asthma of COVID. And it must also have what we call tolerance, meaning that it must also not respond or not do an inflammatory response to something that's harmless. So because there's so many antigens or foreign substances that are passing your gut every day. So you've got food, you've got all of these various kinds of food particles, you've got microbes, you've got all of these stuff. And your immune system needs to be able to distinguish between what is dangerous and what will cause harm over the various different types of harmless substances that will come through your gut every time you eat a meal. And so you don't want your immune cells to be trained to attack everything, because otherwise you would get inflammation and you'll get tissue damage of your gut. And that's bad because that would impair like your absorption and, you know, it'll be bad. But on the other hand, you don't want it to like not respond to like dangerous things. Otherwise you have a risk of getting infected and then causing systemic infection like sepsis or even cancer. Yeah. Okay. And then, so now the next part is, I'm just going to briefly talk about gastroenteritis. So what that means is inflammation of the gut caused by pathogens, basically, that, you know, either you eat in or it's something else. But okay, so basically, fundamentally, all of the microbes or every pathogen needs to follow this kind of 4-step process in order to cause disease. And not all disease-causing microbes can cause tissue damage. So, so there are some bacteria that are able to themselves, they can cause direct damage, either through replicating inside host cells or like just replicating and destroying mucking up with your own cells, or they can produce toxins, which will basically alter our own cells' function and just basically mess it up. But some microbes can also cause damage through our own immune response. And so that's what we call immunopathology. Okay, so I'm just going to go through two types of, two types of bacterial species. So the first one would be Salmonella, and it causes disease without toxins. And it does this by invading our own cells and basically screwing it over, basically. Just to note that only specific subgroups of the Salmonella species here will cause gastroenteritis. So not every single species of Salmonella can cause food poisoning or gastroenteritis. And so there are specific serovars. And so serovars are basically like they're all part of the same species, but it's just like a little subcategory. And we distinguish these little subcategories based on certain features that we see on the bacteria. And this is usually mostly the antigens. So the cell wall antigens and the flagella, or what the other antigens that we can see. These are other species of Salmonella. So not all of them will cause, you know, that food poisoning type. There are other species that might cause more severe kind of systemic diseases. And the transmission, so fecal oral route, meaning that we basically ingest contaminated food that has Salmonella in them, and how it transmits is through our feces. And these are kind of like the common symptoms of, you know, food poisoning. Salmonella is the genus, and Enteridica is basically the species name. Okay, so this is kind of the pathogenesis or the disease progression. Basically, what happens is we eat Salmonella and it's present, it goes through our gut system, somehow survives the stomach, and it gets to the small intestines. And the Salmonella will actually adhere or stick onto certain cells or certain parts of the epithelial cell. Usually we've got like this mucus layer here that kind of protects it, but Salmonella, some of them are able to evade this and kind of beat the mucus layer by secreting certain toxins or certain proteins that can break this mucus layer down. But basically the first step, it needs to bind to these cells. And once it binds to some of these cells, so one of the cells are what we call M cells, and they're present on the Peyer's patches, and it allows the Salmonella to go inside and get access to the inner kind of bit where you've got all of the immune cells sitting there waiting. so the lamina propria bit. And they are able to infect certain cells, so for example, macrophages, and they can replicate inside of them. They're like really OP. They can like prevent, so the macrophages are trying to destroy these guys by eating them up, but salmonella are able to resist that and kind of just hide inside of these macrophages and just stay alive. And so this is one of the mechanisms of how it causes systemic diseases like the typhoid fever before. And so it basically just hitchhikes around infected macrophages, because these infected macrophages are able to move in and out of the stomach. It can go into the intestines, it can move out into the bloodstream, it can go to other places or to like other lymph node places. And because of this hitchhiking thing, this is how it causes systemic diseases. And one of the systemic, pretty serious disease is sepsis, which is basically bloodstream infection. And it can like kind of escape that whenever it wants to. But it likes to hide inside of the macrophages because it's one of the mechanisms that it uses to hide from our immune system. So this is the immune response towards salmonella. Basically, we actually need both of them. We need innate and adaptive in order to clear or completely get rid of salmonella. And this has to be coordinated relatively well. And so how this kind of all works is that the innate immune system is trying to buy time for the adaptive system to kick in, and it's trying to slow down the bacteria from replicating so that your body doesn't get overwhelmed in the first place before the big guns kind of kick in. And so these innate immune cells, or your dendritic and macrophages, will recognize something called PAMPs, or pattern-associated molecular patterns. So these are conserved kind of general structures that you see that you can see across many different types of bacteria. So for example, it's like flagella, something like flagella, because our own cells do not have flagella, but many bacteria do have flagella. So that's kind of a way that innate immune cells can recognize non-specifically between host cells and bacterial cells. You've also got other things like the LPS, or the lipopolysaccharide. So that's like the outer membrane kind of thing that we don't have. And we've got the adaptive immune system here. So those are comprised of B cells and T cells. B cells are your antibody factories. They produce antibodies, and T cells are like the coordinators of both arms, I guess. And they also boost the adaptive and the innate immune system. So these guys so the adaptive immune system recognize what we call antigens, and those are highly specific, and it's dependent on the species. And so these are unique to the pathogen itself. And it's also got memory that it can recognize the antigen if it encounters it again. Okay, so last but not least, we've got toxins. So gastroenteritis can be classified into non-infectious and infectious. So non-infectious means that the person actually eats the preformed toxin. So they don't consume the bacteria or the pathogen itself. They've actually eaten the toxin that was produced by the bacteria, and it's in the food. So we'll go over an example, which is this one. I don't really want to butcher the name. And we've also got another one, which is infectious. And that's the one where we actually eat the pathogen, and the pathogen basically has to survive the stomach and goes to the intestines, and it has to produce the toxin inside of you first, in the first place. And so if you were to guess, a non-infectious basically gastroenteritis will happen quicker. Your onsets of symptoms will happen quicker, within a few hours of food poisoning, and that's because you've already got the toxin and you've eaten the toxin. Whereas infectious might take a little while. It might take like a day or so before you start seeing like the symptoms of food poisoning. And that's because the bacteria has to get to your small intestines, and it has to start producing those toxins. So that takes time. Okay, so we're going through Bacillosaurus. So these are kind of the really quick key features. It forms spores. So spores are like a state of the bacteria where they're resistant to lots of different things, to the environment, really. They're really hardy. They're really hard to break down and remove. And so they're fairly resistant to hot temperatures and acidic pH, so the stomach. And this bacteria is found pretty much a lot of places, the environment, and also food, especially fried rice, because that's often a common source of food poisoning with this kind of bacteria. It causes two types of diseases based on toxins. The first one is emetic, which is non-infectious. So that's the preformed toxin. And then the diarrhea, which is the infectious type. And then these are the kind of the symptoms. Honestly, it's not bad. It's not too bad, but you wouldn't want to have it. It's annoying. Okay, so these are the two different types and kind of how it works. So the emetic non-infectious type, so that was the preformed toxin. The toxin is known as cerulide, and it's a protein toxin. And how it works is once it gets to your small intestines, it's able to bind to these receptors, which are called HT3. These are known as serotonin receptors. And basically what it does is it activates them, and it basically mucks up your cells. Basically, it changes the cell functions. So for example, one of the effects is that it kills the mitochondria off. And because you need mitochondria to survive, you can't do anaerobic forever, the cell will die. It also causes an increased stimulation of the vagus nerve, which I'm pretty sure Stone might go a bit into. It's to do with the parasympathetic system. So that's like the rest and digest system. And it's to do with how fast your digestive system can move through the system, based like food moving through the system. So that's kind of how the emetic types works. And the key side effects is vomiting. And you've got the diarrhea one, which takes a while to kick in, and pretty much instead of eating the toxin, we're eating the spores instead. That's like found in the food. And these are heat and acid resistant. So they can survive the stomach, and that's how they reach to the small intestines. And there are multiple toxins that they can produce. These are just some examples. These toxins, however, unlike this one is like, this toxin is heat resistant. So when we cook it, we have to actually heat it up to fairly high temperature in order to like kill this toxin, this protein toxin. Whereas these ones can be destroyed easily, which is why these aren't preformed, which is why this is a infectious type and not a non-infectious type, because they can be destroyed when we cook it. So yeah. And so the functions of these toxins is basically it causes pores on the target cells. And I'm assuming the target cells are your small intestine, like the epithelial cells. And basically what pores are is it basically just inserts holes into your cells, and it basically just kills the cells. And yeah, so it destroys the epithelial layer. And so you get the leakage of, the substances that are underneath the epithelial layer. So the lamina propria area, like the fluids and whatnot, and they just leak out into the lumen of the gut. And because of that, that's where you get that diarrhea symptom. And yeah, that's about it. I think I'm done.
Oh man, that's a lot of dense content.
I know, right?
Thanks for doing that, dude. That's like I was staring at it. I was like, I cannot remember half this stuff. So guys, yeah, so far, thanks for sticking with us. It's we are, quite a while in. If you need to take like a bathroom break or something, go ahead for a minute. Actually, I want to ask you guys, would you rather I immediately get stuck into mine, which isn't too long, or do you want like a 2-minute break? Say in the chat if you. Stop. Conflicting messages. 2-minute break. I feel most people want to break. All right, so yeah. So yeah, I guess in the meantime, if you have this is a break, just casual. If you have any questions for Lawrence, you can ask him now, maybe as the break. No, yes. Does anyone have any pressing questions? Yeah, no questions then. I think it's all right.
I think I blew them away.
Yeah, dude, it's very detailed. It's a lot of this stuff you guys will it's kind of useful to know, not necessarily now, but if you aren't examined on it immediately, this will be a lot of MCB. So your next semester double subject, that glycolysis respiration stuff, that's like a big chunk.
That's deadly. Yeah.
Immunity is so complicated, cytokines, and oh god. But yeah, you have that to look forward to. But I'm gonna depress you guys before your exam.
Yeah, I don't want to.
I think I'll start mine at 125. So in a little bit. Lawrence, do you want to share screens with?
Oh, did you send me the PowerPoint?
It's on the group chat. It's on the group chat. If you can find it there. If not, I can send it to you again. Other main content will get repeated every year. Oh, TCS. So immunity stuff is a big part of MCB. So your first semester, your second semester's anatomy is pretty different. What else? Yeah, like the glycolysis, the respiration stuff, all of that.
You spent a week on that. You learn about every single step of that pathway.
Every single
It's
step. So everything in
true.
that cycle, you will learn. You'll learn the enzyme. You'll learn how the enzyme's affected by other things. You'll learn the rate limiting steps. You'll learn
It's cool.
Yeah. Is there a lot of chem in MCB? Not really. No. Like you won't know how to do, like you won't have to know how to do like E2 elimination or what you guys are doing on the exam yesterday. But it'll be basic, like you know, rate of reaction, like K-1, K1, like rate constants. That is about it. What enzymes do. So like no more chem. Less chem. Yeah. I mean, you should know. It's very basic, if there is any. So you have that to look forward to. How difficult was MCB?
It's a step up. 6 lectures a week.
It's a step up. But it looks
lectures a week.
It looks like you guys are they're introducing it to you guys a bit early.
Yeah, which is good.
Yeah, not like we didn't. But anyway, like I was kind of worried because you guys had some interesting subjects with the course change. With like this, was it Discovering Biomed?
That was like the first time they did it.
you guys are scared. Don't be scared. It is a double subject, so obviously like you don't have to do another subject. It's like it takes up 2 places. Yeah. It's the main thing is just the fact that like when you're revising, if you don't keep up to date, because it's double the content, right? So it's like double the cram, and it's very integrated. So they like to tie a lot of. I know teachers like to say it, but they actually do tie a lot of stuff from like week 1, week 5, week 7. It'll just be like the one question, and you just kind of need like the big picture. But what would be is HSF harder? Some people find it harder. Some people find it easier. A lot of people hate MCB. Lawrence and I love MCB.
Bro, it's like the best subject. It's better than HSF.
There we go.
Like, HSF in person, like I don't know how you'd survive. Low key, I would fail. Can you imagine?
HSF, if you like anatomy, if you like small facts, a lot of random small facts, and you like HSF. If you want a kind of like understanding of the underlying concepts, and that'll get you through most things, that's MCB. But actually, on that note, I think I'll go ahead and get started. Lawrence, did you find the PowerPoint?
Yes, I did.
Okay.
Share it?
Yes, sir. Thank you. Also, like, let me know when you need to boot, Lawrence. Actually, you can probably just come later. It's fine.
Yeah, I'll probably come later. Yeah.
Yeah, okay. I was get like a feed.
Okay, I need to share my screen, right?
Yes, sir. Is it enabled? Okay, here we go. Yes, let's go. Nice. All right. I don't think you guys went into too much detail with a lot of this stuff, so I won't go into that, especially given the time. Homeostasis. What is this? This is maintenance of stable internal conditions around a target, and they call it a relative set point. The main thing to say here is the fact that like, well, your body needs stable internal conditions to survive. I think they mentioned cells are protein factories, right? So, you know, proteins play a big part in our biology. If you change the temperature, pH, you know, charge in the cell, that can interfere with protein structure, protein folding, and then that will change protein function, which is, you know, one of the primary reasons why we need to maintain all these parameters in our body. Now, they always like to ask, like, is homeostasis, you know, just remember it's dynamic. So it's not like a constant set level, but it's a constant oscillation within a range. If you fall outside that range, you get to disease. But yeah, I mean, the other thing is, yeah, just reflex pathways. So as you see on the slide, you'll have an input, a stimulus, you know, there'll be some sort of imbalance, too much, like too low blood pH, right? You'll get a receptor detecting that, a bit of control center, effector, it'll correct the stimulus, and you're kind of, you know, it'll balance itself out. Lawrence, next slide. Thank you. All right, so now with those feedback loops, there are 2 types. There's negative feedback, positive feedback. Negative feedback, closed loop. They all look like circles and closed loop, so just ignore that. But it's called closed loop because it corrects itself, right? So whatever stimulus you have will evoke a response eventually, which mitigates that stimulus. Whereas with positive feedback, it's amplification. It's an open loop because you'll have a stimulus that evokes a response, which in turn increases that original stimulus, which will, you know, increase that response, and it will loop on, and it'll have a bigger and bigger response and stimulus until you have an external thing to switch it off. The key thing being that with negative feedback, you don't need an external like off switch. With positive feedback, you do. Next thing, Lawrence. Also, if you need me to slow down at any point, just say in the chat and I'll slow down. Negative feedback. So this is they like to ask you like, oh, what's the stimulus in this pathway? What's named the, afferent pathway? What's the integrating center? We had an exam a few days ago, which had a question like that. Maybe that was a practice question. I don't know. Anyway, negative feedback. So you start with a stimulus. That's the change, such as too low. Actually, we'll go through an example later. But so stimulus is the change. You have a sensor detecting that change. Afferent pathway is the kind of what's relaying the signal from the sensor to the control center, that integrating center, right? So you have the sensor, you have the afferent pathway, and then you have the integrating center. From the integrating center, it'll compare, it'll integrate the response. It'll say like, okay, this is too much of this stimulus from this pathway. I need to correct that. And it'll send an output via the efferent pathway. So that's the outgoing pathway to the effector, which is like whatever is producing your response. Your response will go on to, what your response is to change the original stimulus, to decrease it or mitigate it. And then, it kind of balances itself out. It'll naturally kind of taper off. Next slide, Lawrence. So quick examples, thermoregulation. Only go through like half of this diagram, just because it's like the same thing. But if you had body temperature too high, temperature sensors will detect that, right? You'll have the integrating center in your brain somewhere, as a hypothalamus perhaps, maybe, I don't know. Your integrating center will realize like, okay, this body temperature is too high. It'll send signals via the efferent pathway to your effectors, which in this case, you have blood vessels in your skin. Those will dilate, so you have more blood flow to your extremities, to your skin, where heat can be lost. I guess you'll also sweat. You might, there's some behavioral changes. You might seek shade, etc. Anyway, so at that point, you have more heat loss. Your body temperature will go back down. Your stimulus will, kind of correct itself. And there you go, negative feedback. Next slide, Lawrence. Positive feedback. Childbirth, parturition. In HSF next year, this is the whole thing. But essentially what you have is like the baby, its head will push against the cervix. That is the stimulus, right? Your body will detect that. It will cause more secretion of oxytocin, sorry. That will cause a stronger contraction, which will push the baby's head more into the cervix. So you have more. Lawrence, do you want to take the chat, by the way, while I'm doing this? But yeah, so yeah, oxytocin will have more contraction push the baby's head harder. You'll have more oxytocin. Obviously you see how this kind of loops back and back until the baby is ejected. Now, I mentioned before that you need an external switch off, in which case for this, the baby leaving the body is the off switch. Next slide. Here we go. All right. So now a bit of a jump to nervous system stuff. I won't go too much into detail here, but the broad strokes of kind of the fundamentals of what you need to know here are that somatic division of the nervous system is kind of like what's conscious, right? It's like I can hold my breath, I can lift my leg up, I can decide to. I don't know, something else. But like, I'm in control. I'm in conscious control of these things, right? So that's the somatic nervous system. The autonomic stuff, autonomic division of the nervous system, that goes to your, your viscera, your organs. I can't control my my blood pressure. Well, actually, like I can't control the level of vasoconstriction of my blood vessels to regulate my blood pressure. I can't do that consciously. My body is automatically doing that. That's the autonomic division. Now, the autonomic division is subdivided again into two further components, the parasympathetic and sympathetic nervous systems. You guys probably heard by now, parasympathetic is like rest and relax. Sympathetic is fight or flight. Actually, next slide, Lawrence. So yeah, fight or flight. It's a little like that covers most of it. And like, I'll give you a few examples. Parasympathetic, if you're resting and relaxing, right? You eat a meal, your parasympathetic system will become more dominant than your sympathetic. Your gut will become more motile. So the smooth muscle in your gut will contract more, I guess, and you'll have a more active digestion process. In contrast, when you are, when your sympathetic division is activated in fight or flight, so you see a lion or something, you're running away, your gut motility will decrease because you don't need to do that digestion right then. However, it's a little more complicated than, I'm just reading the chat. Yeah, so it's a little more complicated than one being fight or flight, one being rest and digest, because sometimes they do work together. For example, and I guess get comfortable with this now, guys, because when you do HSF, there's a lot of reproductive anatomy. With the male ejaculatory response, right? When they use the point and shoot. Yes, they use the point and shoot. Like I don't know phrase to remember it, but your parasympathetic system is used to activate the erection. The ejaculation phase is by your sympathetic nervous system. So you have a response here where your two nervous systems aren't, one's not really, you might think, oh, that, that arousal is fight or flight kind of excitation, but it's actually both. The other thing to note is that in your body, you'll have a tonic level of activation of both of these things. So you'll have some parasympathetic activation, some sympathetic activation. It's an overall balance, though, between the two. That determines what is going on at that moment. So, but the main thing there being the fact that one isn't just on, one's not off. It's a balance. Next slide, Lawrence. Okay, now again, jumpy content because I was just picking out what I can help you guys with. Neurotransmission. Actually, no, this does link with it because it's the somatic, autonomic, sympathetic, parasympathetic stuff. I won't go into too much detail here, but the main thing being with the somatic nervous system, right? That's like, conscious control of my contraction of my biceps. You'll have a single neuron going from your spinal cord, single motor neuron. Cell body there, it'll go all the way to your muscle. It'll release its neurotransmitter. And we'll talk about neurotransmission in a second. It'll release this neurotransmitter at that site in the muscle, cause a contraction. Parasympathetic, the neuron goes pretty close to the site of to the target tissue. There is something called a ganglion. Yes. Yeah. So you have a preganglionic and a postganglionic neuron for the parasympathetic and sympathetic divisions mostly. You'll see how there's like those two in series, right? And the preganglionic one is the one before that kind of synapse between the preganglionic and postganglionic neuron. But for the parasympathetic system, the preganglionic is very long. It'll go very close to your target tissue. Your postganglionic is very small. That's that. With the sympathetics the other way around. You'll have probably don't even know this, but sympathetic chain really close to your spine. And your preganglionic is very short, goes to that chain. Postganglionic is very long. It'll go towards target tissue. There is some complication with the parasympathetic system, because you have chromaffin cells. It's not like two neurons in series anymore. It's one preganglionic, not really, neuron going to that chromaffin cell, causing adrenaline release circulation. Next slide, Lawrence. All right, so I saw this in your slides. Very briefly say this. Hormones, endocrine cells. Hormones can also be secreted by neurosecretory cells, which I do think are neurons. And then you have neurotransmitters also released by neurons. Hormones circulate in the blood. They act far, like the whole body system can act at a distant site compared to the endocrine or neurosecretory cell. Nerve signals, neurotransmission, that is very local, right? Your neurotransmitters will diffuse very short distance across the synapse, act on the other neuron or the other type of target cell. Next slide, let's. All right, sorry for the low res pictures, guys. This is just screenshots of my lecture recordings. But so you have I won't go into each pathway here, but basically you see the stimulus, you see the receptor, the afferent neuron, the one carrying that response to the kind of integrating center, in this case, the CNS. Some cases you have that neuron releasing, hormone into the system. Sometimes it will synapse, it'll have neurotransmission with an endocrine cell, which will then release a hormone. Sometimes you have the neuron releasing a neurotransmitter, goes to the endocrine cell will make a hormone to another endocrine cell, which will then go to your target tissue. Very roundabout, but it can get complicated, right? Which is the bottom line. Next slide, Lawrence. Okay, now this is kind of like a 180. Fertilization. Not too much to say here. You guys probably know more about this than I do, but there is a structure in the cap of the sperm head, the acrosome. Upon joining with the egg, the oocyte, you'll have some acrosomal reaction, some digestion of that. It'll help the sperm go through to the egg. Once they're joined, you have fertilization. You have some sort of cortical reaction, which will prevent other sperm from entering. If you do, you can get these kind of twin situations, kind of weird. But anyway, that's that. Next slide. Ovulation. Okay, so yeah, this by the way, this entire section, following section, is about the whole kind of embryonic development overview stuff. But yeah, if you see here, you have the ovulation, the oocyte exits the ovary, gets fertilized successfully by the sperm, usually in the somewhere in the oviduct, I think. And then anyway, so after it gets fertilized, you have a series of divisions, cleavages, we'll talk about in a second. You get a blastocyst. The blastocyst will implant. You get your embryo developing. Next slide, Lawrence. Cleavages. Only thing to say here is, and you guys probably know this, but cleavages there's no cell growth between cleavages, right? So they will increasingly make each daughter cell smaller until you get this little mulberry, right, called the morula, which is a little clump of little cells. Next slide, Lawrence. Thank you. Blastocyst. All right, so once you have the morula, you have cavitation, so you have fluid being pumped in, right? You'll have an outer layer of cells called the trophoblast, and an inner layer, an inner cell mass, which is what it sounds like, right, on the inside of that trophoblast. And then you have a cavity as shown in the diagram there. The preemptively change slides. The trophoblast forms extraembryonic structures, so it'll form like placenta. It gets a little complicated, but the ICM, crucial point to note here is the ICM, that's gunk in the middle, is what actually forms the embryo. That's what actually forms you. Next slide. Yeah, so the ICM itself, so there has been differentiation to get the trophoblast and ICM, but now the ICM further differentiates and you get something called, well, you see in the diagram, the epiblast and the hypoblast, right? So you get these two cavities forming within the ICM. There's the cavity outside of that, so I guess there's 3 cavities total right now. But I guess kind of we're following through where the embryo actually comes from, which would be, in this case, the embryonic disc, which is that epiblast, hypoblast. So that's that kind of section in the middle you see, right, with those 2 layers, or else can outline it with is. It's just the disc though. It's just the hypoblast and epiblast, but that embryonic disc is where the embryo does come from. Next slide. Gastrulation. You guys did learn this. This is the formation of those 3 germ layers. So it starts with the primitive streak formation. So that groove forming on the epiblast, I believe only the epiblast, so only that top layer of the embryonic disc. Once that streak, apparently it's thickened cells, once that streak is formed, you have the definition of body axis. So as soon as that streak is formed, you know what's top, bottom, left, right, head, tail, essentially. I won't go into detail there, but it's on the diagram. The groove forming on the epiblast, which is on the top, I guess you imagine like the top side of your body, that will define, well, the back rather, for lying down, but that'll define the dorsal side. So it's like kind of your back. It's like the fin of a dolphin. Once you have that, then you, based on where the groove extends, what's head, what's tail. So the groove will not go all the way to your head. So you'll see that gap. That's the cranial side. Caudal, kind of your tail, bottom section. Then you have left and right, consequently. Once you have the primitive streak, that will deepen, form the primitive groove. Next slide, Lawrence. Then we have cells migrating through the primitive groove from the epiblast, so that kind of top layer, right? They'll first go into the endoderm bottom, which is the kind of like the hypoblast layer. They'll migrate in there first. After that, they'll migrate into the middle between the bottom and top, so that's the mesoderm. And then the cells left at the top will be the ectoderm. Next slide. This is just to kind of recap. I know I've been going quickly through this, so thanks for bearing with me. But if you look at the diagram on the left, you'll see that the 3 germ layers are responsible for just every tissue organ in your body. The only exception being the germ cells. So your sperm and eggs are set aside before the formation of the 3 germ layers. If you look on the right side of the diagram, there's the whole overview about what forms from what thing. If we look, so you see the trophoblast, it forms some, don't worry about the terms here, but some extra embryonic structures. In your cell mass, you get the 3 germ layers forming from that. The very end of the diagram, that's where your embryo is from. All this other stuff is like the accessory structures, right? Your yolk sac, all these things will go away eventually. Next slide. Not a chord. All right. I really like this. It's not a chord for some reason, but what you have is you have that primitive groove, remember, and at the end of that groove, you had like a little node, and cells will go in that little hole at the end of the groove, and they'll migrate in. They'll form a tube, going in the direction you see there. This is a transient structure, right? So it doesn't form anything permanently, but it's there to induct the neural tube, which is very important. Next slide. All right, neural tube. This is how you get there. So once you have the notochord underneath that top layer, the cells above it will be signaled to thicken. They'll become the neural plate. That neural plate will then curve in more. You get the neural groove, and then once it joins over, that's the neural tube, which forms the central nervous system. Next slide, Les. All right. Upon that joining, you have this kind of like the cells of the joining bit, they'll migrate away. Those are the neural crest cells, important for various things I won't go into. You'll learn about that more next year. The side note, interesting note here is that these cells undergo EMT, right? So they go from an epithelial type to a mesenchymal cell type, migratory. So they go from kind of like that polar apico-basal polarity. So they go from a defined organized structure to something that's more, free flowing. This is a very important process in cancer, which is a huge topic for MCB. It's like everything they do is pretty much around the bottom line of cancer. But yeah, this is a big process in how cancer can metastasize, right? So it'll form somewhere in your body once it undergoes EMT, migratory, go into your blood, go elsewhere, and then you get metastases, which are what secondary tumors, dangerous stuff. Next slide, Lawrence. All right, brain. Again, really quickly. Okay, so left and right hemisphere, the main groove between the left and right hemisphere, like that vertical line in that graph, yep, longitudinal fissure that separates the hemispheres. There's stuff called sulci, singular sulcus. Those are the grooves, and the peaks, if you will, the bumps. Those are the gyri, right? So between your gyri, you have between each of two gyri, you have a single sulcus. Hills and valleys. This is to increase surface area for the brain. More neurons. Your brain obviously has a lot of neurons. What else? Oh, cerebellum. Yes. So the little back bit of the brain. Yeah, that little weird looking thing at the back. It is the cerebellum. It has a lot of neurons too, and it's very important. It has its own anatomical names for things like the gyri are not called gyri. I think they're called folia, Lawrence. Do you remember? Something. Anyway, you probably don't need to know that. Next slide. That's an overview of brain structure. The brain is divided into regions where, each region associated with some sort of function. You have the precentral sulcus, the postcentral sulcus in and around the central sulcus being so you had the longitudinal fissure down the middle. Across left and right, you have the central sulcus. The kind of the bumps in front of and behind that are for different functions. I think the one in front is for somatomotor coordination, so movement stuff. The one behind it, the postcentral gyrus, is for sensory input. And you'll notice for next year a lot of stuff, which is a lot of stuff in the CNS in the front is for somatic, control of skeletal muscle, right? Stuff in the back is sensory. It's just an odd little note, but it'll help you. Next slide. All right, so actually, go back one slide. Sorry, I finished. There's association areas. So these areas have different roles. You'll have other areas nearby which will have roles which seem to be associated. Makes sense, right? They're close by. So just note that there is some organization to the way the brain functions. Cool. Next slide. Brain, corpus callosum. All right, so this is the kind of bridge section between the right and left hemispheres. If you look at the colors in the actual thing, but you have white and gray matter. So white matter is white because it's myelinated, right? So you have learned about neurons. Some of them are wrapped in myelination. It's kind of like lipid coating. It's insulatory. It helps signal conduction. But essentially, if you look at the brain, the inside you have white matter, outside you have gray matter. In this case, it makes sense because if you want communication between the left and right hemispheres, you want that bridge to, it's communicatory, right? It's bringing signals across. So that's therefore white matter. The cell bodies are unmyelinated, so they're gray matter. They're on the outside generally. The spine is a reversed conformation of that. So you have white matter on the outside, because if you think about it, you're sending nerve signals from your spine out to the rest of your body. The inside will be gray matter. Anyway, next slide. Brain, ventricles. All right, so brain ventricles, little compartments. You guys learned about CSF and production, but essentially what you have is you have these little capillary beds called the choroid plexus. There are multiple. I think there's one in each ventricle, each brain ventricle. And these capillaries are fenestrated, right? So they have little holes in them. When blood filters through, a little bit of the plasma and stuff in the blood will filter out. That filtrate is going to form your CSF, and it'll go around this system of connecting networks and tubes, and eventually it'll enter the subarachnoid space. Not really go into that because it's not on the slide, but kind of this outer area just bathing the brain. It's like a cushion. It helps protect your brain. Next slide. It is reabsorbed. If you look there at the top bit, the arachnoid villi, right? So this is a little projection into a big blood vessel there called the something sinus, the sagittal sinus, there you go. And so this fluid, the pressure will build up as CSF is produced. It'll filter across into that blood, and then you have the net filtration reabsorption being balanced, so your brain's fine. If it is not balanced, you can see what happens to the baby's head. It looks like a watermelon. Not too dangerous because pretty dangerous, but the kid is okay with a minor operation to kind of pump the CSF out. But anyway, that's just a point on that. Next slide. Okay, this is the last section, guys, so we're almost done. Receptors. Different types of receptors. Ligand gated. So ligand will bind the receptor. I think they are ionotropic, which means they're like ion channels, right? So once a ligand, such as acetylcholine, binds the receptor, it'll open up. Your channel will be open. Ions can go through. Because it's ion flow, it's very fast. This is the kind of depolarization. Actually, no, that's voltage gated. Scrap that. So we'll talk about that in a second. GPCRs, metabotropic. So yeah, so these can be either ionotropic or metabotropic. We just talked about ionotropic, so that's fast, ion conductance. But metabotropic takes a little longer because you need a whole system, a whole chain of reactions, a secondary messenger cascade in order to do some stuff to the cell. Anyway, it takes time, right? So you have all these steps, so it takes a little longer than your ligand-gated ion channels. You guys, I won't go into it, but you have G proteins bound to GDP. If they're GT, switches to GTP, is active, diffuses across, does some stuff. Anyway, next slide. I wonder who's, but Lawrence, can you do the next slide? Yeah, there you go. Kinase linked receptors. All right, so this is the third type. Something binds, the ligand binds, you have transphosphorylation. The inside's phosphorylated, you'll have something coming across. I won't go into the complication there, but essentially the main thing here is you have gene transcription changes. You have which take time, right? Because you have this whole process going on, then you're supposed to transcribe genes differently, you have new proteins, different proteins that will produce an effect, but it can take a long time, adding hours to days. It just says hours on the diagram, but when it comes to nuclear receptors, kind of same principle, right? Something will diffuse, and this time it won't bind at the plasma membrane. It'll go through into the cytoplasm. It'll bind in the cytoplasm. That receptor ligand complex then will dimerize. So like two of them will come together. It'll go into the nucleus. It'll interact with DNA either indirectly or directly. Don't ask me what that means, but it will interact with the DNA, and then you'll have changes in gene transcription, protein synthesis. Again, because of that, it'll take some time. So, if you look in total in the diagram comparison, you have ionotropic on the very fast side, and then kinase linked and nuclear receptors on the very slow long-term side. Next slide, Lawrence. All right, so cells of CNS is very brief, but you guys know what neurons are. Conduct signals. They're the functional units of communication. Glial cells. These are like the supporting cells, right? Not just like architecturally supporting it. They have a bit more of a complicated role, but a few types are listed on the slide. You got astrocytes. You have, they help synaptic transmission somehow. I don't think they taught us that. Oligodendrocytes, which form myelin, so that forms that kind of fatty coating, right, which helps us.
Now arriving at stepping.
Cat, I think you gotta mute yourself. I'm assuming that's Cat. And then you have oligodendrocytes. Yeah, so these can myelinate multiple neurons per oligodendrocyte. There's something called a Schwann cell, which is in the peripheral nervous system. It's not in your brain or spinal cord. That will myelinate one neuron. Anyway, extraneous detail. Microglia, apparently immune cells. Next slide. Neurotransmission. Okay, I won't bore you with this. You know what they are. You know, you have dendrites that kind of where the input from other neurons or sensory input will go into the thing. It'll be integrated. Your soma, your cell body. It's like the stuff, the basic housekeeping, keeping the cell alive. The axon, the important part, that's the long transmission conductance there. Can be myelinated, doesn't have to be. Then yeah, it goes all the way to your nerve terminals at the end, which is what we will look at in the next slide. Next slide, Lawrence. Yeah. Okay, there you go. Actually, I lied. It's like 2 more slides. But so cells in the CNS. You have the axon hillock, which is the region joining the cell body to the axon. You have the axon itself, that long bit, and then you have the terminal at the end, right? These 3 places are the only places where you have voltage-gated ion channels. Important because these are the only three places you can have an action potential traveling. Yeah, next slide. Okay, so next year you'll go through each of these steps in a lot of detail. But just a little overview, resting membrane. So you start from resting membrane potential. No voltage gated ion channels are open yet, right? It's all leak channels. So these are things which don't need membrane potential to trigger. They're just constantly always open. You have some ions leaking in, leaking out. There's pumps, there's various things, but this is the starting state, right? And the important thing to note here is that resting membrane potential is negative. So you're starting with the inside. Remember, the inside, not the outside, the inside of the cell membrane. Well, inside of the cell is more negative than the outside. When you have depolarization, so depolarization is what it sounds like. If you start more negative, you want to depolarize, you want to unpolarize it, right? It'll go towards 0. It'll go up from negative. So that's the depolarization side. Repolarization will be going back down. So polarizing again, making negative again. Then there's hyperpolarization. It's too much polarization, too negative. So that's like the dip below resting membrane potential. But so yeah, we'll look at each of these steps starting from depolarization. So what happens here? At the due to various things such as excitatory, postsynaptic, something, due to various triggers, right? You'll have the membrane potential becoming more positive. So it starts negative going up. If it reaches threshold, then you'll have action potential generation, right? So if it reaches that threshold, you will always have an action potential then subsequently generated. What happens is these sodium channels, these voltage-gated sodium channels, so they're sensitive to this level of depolarization, right? Level of the membrane potential. At a certain point at threshold, a bunch of these all of them will be triggered to open. You get sodium going into the cell due to gradients and stuff. We'll talk about it. But what you have is positive charge going into the cell, right? Which would make the inside of the cell, you'll make the inside of the cell less negative, therefore depolarizing it. Next slide, Lawrence. Repolarization. So potassium channels are a bit slower to open, and they'll open at the peak, right? The peak of this membrane potential graph. Your sodium channels will close at this point. They'll be inactivated. There's a whole complicated steps. The sodium channels have interesting architecture. They're like inactivation gates and stuff. Won't talk about it. But so yeah, at the peak you'll have the potassium channels finally opening. You have more potassium on the inside of your cell. It wants to leave the cell through reasons you'll find out next year. And when these voltage-gated potassium channels are open, right, you have potassium leaving. Your sodium is not entering anymore because the thing's closed. And so yeah, you have positive charge, the potassium leaving the cell. So the inside of the cell is becoming less positive, more negative, therefore repolarization. Hyperpolarization comes about because these voltage-gated potassium channels are slow to close. So they're open for a bit too long. More potassium will leave. than required to get to that resting membrane potential. It'll overshoot and you'll be more negative than you started out with. So that's the resting, that's the hyperpolarization phase. Next slide, Lawrence. Now, through all this stuff, so what we just talked about was the conduction of an action potential, right? That's how, anyway, so once your action potential goes all the way down the axon and reaches the nerve terminal, if you look at the graph, you see it coming in at the top, it will cause the opening of voltage-gated calcium channels, right? These calcium channels will cause the exocytosis of synaptic vesicles. So these little packages of neurotransmitter waiting at the nerve terminal to be released. These have been formed beforehand. So if you look over there, there's like synthesis storage steps before. But essentially what you have is these synaptic vesicles waiting at the nerve terminal. They're just right at the edge. Once the action potential comes, it's triggered, calcium goes in the cell. There's various stuff that happens. And then this synaptic vesicle is exocytosed, so it's released. The neurotransmitter goes across the synaptic cleft, binds some receptors on the other side, will cause, and so this will probably be at like those dendrites, right? These ligand-gated ion channels will open at the postsynaptic membrane. You'll have ions going in or out. You have a change in membrane potential. If you have enough of that, you'll trigger another action potential at the next neuron. Next slide, Lawrence. Oh yeah, so I think this is like the last slide, so you guys made it. L-DOPA. This is kind of to do with what I saw in your slides, something called a dopamine hypothesis. But just we're quickly looking at how dopamine is generated, synthesized, how noradrenaline is synthesized, and how adrenaline is synthesized. It's all one pathway. Not necessarily taking place, like some cells only go to noradrenaline, some cells go all the way to adrenaline. But anyway, you start with L-tyrosine. There's tyrosine hydroxylase enzyme converted to L-DOPA. There's dopa decarboxylase, convert that to dopamine. It's pumped in through vesicular monoamine transporters into the synaptic vesicle, where this enzyme called dopamine hydroxylase acts to convert dopamine to noradrenaline, which will then exit the synaptic vesicle if this is in the, I think the adrenal glands, the chromaffin cells, and then that PNMT enzyme, which we never learned what it's, you don't need to know what's like the full name, that will convert noradrenaline to adrenaline, and then that can be exocytosed, right? So yeah, this is just for each step, and also actually, I'll say this and we'll go back to the previous slide in a second, not yet. For each step of this pathway, right, you can have drugs acting to inhibit some enzymes, to inhibit some transporters. Anything can be modulated to change the response of our body to these different neurotransmitters. So let's say if you don't want, if you inhibit the transporter which moves dopamine into the synaptic vesicle, it'll stay in the cytoplasm, it'll get degraded by cytoplasmic enzymes. Maybe you'll have a decrease in level there. If you pump in more L-DOPA, right, you'll have more synthesis of dopamine. That's one of the treatments for either Alzheimer's or Parkinson's, one of those two. So like just remember, like the law, a good way to revise is to think about a pathway and think about what can go wrong and how that will impact the pathway. A lot of times with questions on exams, certainly next year, possibly this year, they'll ask, okay, this is the pathway. Let's say this step, this enzyme is hyperactive or it's not as active. What's the consequence, right? But it's a good way to consolidate your understanding. Go to previous slide, Lawrence. Same thing here. So there's a lot of steps here, right? At each of these steps, you can use a drug to modulate what goes on. You can say, okay, let me block the voltage-gated channels, ion channels, so there won't be action potential propagation. You might get some sort of paralysis. You could increase synthesis of a drug, of neurotransmitter, sorry, so you have more neurotransmission. You could inhibit that synthesis, so not as much. You could, there's Botox, Botox like cosmetic surgery, that can inhibit the exocytosis from occurring. So it does some cleavage stuff, don't worry about it, but it can prevent these neurotransmitters from being released. So you get this kind of local paralysis. That's why, no wrinkles, right? There's, you can change uptake. So I didn't talk about this before, but once the neurotransmitter is released into the synaptic cleft, it can be uptaken into either the nerve terminal or presynaptic nerve terminal or the postsynaptic thing. It can be degraded more. Anyway, you can change each of these steps and you'll have a different response. Okay, with that, Lawrence, next slide. This is the last slide again. Last thing to say is just the monoamine theory of depression, just the fact that neurotransmitters, there's a chemical basis for depression, basically. Different levels of neurotransmitters can be influencing mood. So that's it. Yeah, and with that, I think that is the last slide. Lawrence, yep, that's it. Thank you everyone for staying so far. It's a long one, but there is a lot of content. And yeah, again, I just want to thank Lawrence and Kat. I think Kat's back as well. And thank you to everyone for listening to us. Hopefully this is useful. There might be some details which were a little extra over what you guys learned. Maybe some other stuff we didn't cover because we haven't learned the exact same course. But yeah, hopefully it helps. I think Lawrence, actually, if you can find the last slide Kat did, which is about exam tips.
Did Kat put her slides on the thing?
I did, yeah, but I can just go through it. Yeah, if you guys can hear me.
Yeah.
I was just going to say, basically, with your exams, just try to basically do a lot of practice questions in the lead up. It's easy to revise content, but you won't process as much of it as if you did practice questions. That's my opinion personally. Sorry if it's a bit loud, guys. And then the only other thing I was going to say was just during your exam, first year subjects are pretty notorious for being long exams and it being hard to manage fitting in all the questions. So I would just say if you get towards the end of your exam and you're feeling like you're really pressed for time, just at least pop some dot points on those questions just to get the main ideas out. And that's basically what will help you to secure the H1. Because you just have to show the lecturers that you know what you're talking about. You were going to say something here rather than just leaving like an 8-mark question blank, if that kind of makes sense. But otherwise, feel free to message any of us with any questions that you have. Feel free to message BSS with any questions, or even if you're feeling stressed for exams, we're always here to support you guys. So yeah, I'm wishing you all the best of luck.
All right, I think Lawrence is gonna go. So that's, Lawrence, you're free to go.
Yeah.
Also, yeah, after exams, guys, come to EOX, come to BSS Cruise. We have, it's gonna be good. It'll be good. But anyway, there's a lot of pressure now. It's a great way to relax. So that's there for you guys. I think everyone has to go pretty much now because we have a separate meeting to kind of attend. But yeah, that's about it. Hope you guys enjoyed it. Bye.
I think this will be uploaded as fast as possible.
Yeah, it took a day though because it's like big upload.
Yeah.